I'm Kirtana. Our project title is Identification of Genomic Rearrangements in Genomes Leading to Evolutionary Insights. So here we'll be looking into the large structural changes in the genome like deletions or inversions and understand how they have a contribution to the evolution. So the common genomic rearrangements such as inversions, duplications and deletions, they can significantly influence how species evolve. So here we'll be detecting these rearrangements between the different types of genomes using comparative genomics for alignment and comparing the genome structures, and then the phylogenetic analysis to understand the evolutionary relationships. So these are the contents, introduction, objective, dataset collection, tools and technologies used, methodology, current status, results and visualizations, chromosome length comparison and DNA and similarity plot. Then the further improvements, summary and references. So even though humans and chimpanzees, they share about 98.5% of the DNA, there are still clear differences in how they look and we look and behave and function and all. So these differences often come from genomic rearrangements and the structural changes and all can affect how genes are expressed, and it can lead to a big major evolution. So here in this project, I'll be using some bioinformatics tools and Python scripts to compare the genome sequences. So the main goal of the project is to identify and analyze the structural rearrangements and study their impact on genes and regulations and all. So genomic rearrangements are big changes in order of DNA. It's like it includes all these inversions, deletions, duplications, and translocation. Inversion means a piece of the DNA is flipped, and deletion means some part of the DNA is lost. Then duplication means it's like a copy is there, and translocation means some part is moved to another part of the chromosome. So we are trying to find all these differences. So here we'll be collecting the genome data from datasets we'll be downloading from the site websites, and then we'll do the pre-processing. Then we'll detect the big structural changes, and then we'll understand it. So here I downloaded two files, FASTA files and GFF3 files. So FASTA files is used to work for it contains raw DNA sequences, and GFF3 files have the information about where the genes are located on the chromosome. So methods, first dataset collection, I downloaded genome sequences for human and chimpanzee from Ensemble, and genome data, FAST files, and gene annotation, GFF3 format I downloaded. Then we did the structural comparison. We compared the length. Then we extracted the base pairs of the genome and then compared it between human and chimpanzee. Initially, I used sequence matcher in Python, but it had a very small similarity. So then afterwards, I use pairwise sequence alignment. It could give more accurate results. Then another method I used was sliding window comparison. Here it will like detect in a window with a definite size. It will detect which all genome is same or similar between human and chimpanzees. Similarity scoring, we calculated the percentage between the similar things and we visualized it. So current status, I downloaded the data sets and from the public databases such as Ensemble. So I did the pre-processing and data handling using BioPython and then started the comparison of chromosomes, compared the chromosome lengths between human and chimpanzees, and then visualized the result using a bar chart. Next is the comparison based on chunk based. Like we'll extract 1,000 base pair chunks from the first chromosome, and then with this in the same position of the chimpanzee genome, we'll check if it is similar. And using sequence matcher in Python, I did this. Then I did pairwise using BioPython pairwise. This is more accurate, and we got a percentage of 42. The sequence similarity visualization was done. We plotted it on a graph, percentage identity, and we could see some high and low areas.
Okay, so we can look into the code section. Here, this is a compare genome code. Here, we compare the size of each chromosomes in base pairs from human and chimpanzee genomes. So we are loading and then reading both the genomes. Then we compare it. It's a simple method. So here we are. This is the output. Here we can see this chromosome 1, chromosome 21, 22, then 9. All these are huge numbers. So the difference is very high. So we can say that there's a possibility of mutations such as deletion, duplication, and all. So we can infer. Before that, we also did pre-processing the data and we read the human chromosomes. So this is a code for that. So in this we open the human genome GFF3 file and we read it line by line and we print the gene IDs with chromosome number and start and end positions. So this is a part of the output. I didn't include the whole output here. I ran it in WS. That's why I added it in comment. So this is the another method using BioPython's pairwise to calculate the similarity. It gives a more accurate result than the other one. Other one is a simple method. So here we are loading the chromosomes and loading the chromosome 1. We are only checking with this one for the timing. Then we are extracting 1,000 base pairs from the starting position. This is the starting position and we are avoiding the missing values and all, and then we are comparing, and this is the output we get. So score is 592 and the percentage is 42. So this is actually low compared to the experimental this one. So I will be doing it in the like further improvements. Then we plotted the graph. So this is the code for the graph. I will be showing the graph in the further slides. This is the slide compare method. Here we are loading the chromosome, then checking the similarity. So this is a window sliding method. So chunk size is 1,000 base pairs, and we start at this position, and with the window size of this uh, range, we search the chimpanzee region, and we get the result is also there in the further slides. The next, this is a passing code which was used earlier. So here we open the human genome, the GFF3 file, and then it reads line by line and then prints the gene IDs with chromosome numbers and start and end positions. So this is the output. Chromosome numbers. This is only a little part of the output. I could only include this much. It's a big this one. The next read genome. This one also I did initially when we are pre-processing the data. So it just reads the chromosomes and the length of human genome. Okay. Then we plotted the chromosome lengths of humans and chimpanzees. The result is also there in the further slide. Yeah, that's it.
So this is a graph which compares the lengths of chromosome between humans and chimpanzees. As you can see, they are very similar in structure. So that from there it comes our shared evolutionary history. But there are subtle differences between them. So from there we can understand the different distinct traits which we have between us and chimpanzees. So this graph shows the percentage of genetic identity between humans and chimpanzees when we select for chromosome 1. So the peak areas are the regions of high similarity, and the down deep shallow areas are the regions of low similarity. So till what I have done, I only used pairwise 2 for the small chunk comparisons, but large tools such as LAS, TZ, or BLASTIN can be used for better because the data set is very huge. And when coding, I avoided the unknown basis n to avoid the noise. So that also should be improved. Then I only compare the matching positions. So some places it will be inverted or something like that. So that also I have to improve. So here, till what I have done, I collected the data, then pre-processed it, and then tried some manual methods to see the similarity between them. And I will be uh, going into deeper research to know about all the stuff and to get more accurate results. These are the references used. Thank you.